First, the epidermal growth factor binds to the extracellular domain of an RTK on the cell surface. The body releases growth factors in response to signals like skin injury or the need to replace old cells. The ligand binding causes two RTK molecules to come together to form a dimer. Dimerization is necessary to activate the RTKs. This step ensures the signal is amplified. The intracellular tyrosine kinase domains of the dimerized RTKs phosphorylate each other on specific tyrosines. Phosphorylation creates binding sites for downstream signaling proteins, converting the extracellular signal into a molecular signal inside the cell. Proteins like GRB2 bind to the phosphorylated tyrosines on the RTK. GRB2, in turn, recruits SOS, a guanine nucleotide exchange factor. This step acts as a bridge between the activated RTK and the next component of the pathway, ensuring proper signal relay. Activated RAS initiates the MAPK cascade. This involves a series of phosphorylations. The MAPK cascade amplifies the signal and delivers it to the nucleus, where it can influence gene expression. Phosphorylated ERK enters the nucleus and activates transcription factors. This step directly impacts gene expression, turning on genes involved in cell cycle progression and new cell synthesis. Transcription factors activated by ERK bind to DNA and promote the expression of genes like cyclins and CDKs, which drive the cell cycle. During prophase, chromosomes condense and become visible, and the nuclear membrane starts breaking down. Spindle fibers begin forming from centrioles. This organization ensures chromosomes are ready to be accurately distributed, preventing errors that could affect skin cell function. In prometaphase, the nuclear envelope fully disintegrates, and spindle fibers attach to chromosomes at their centromeres. This attachment is crucial for guiding chromosomes into proper alignment, ensuring each new cell gets the correct genetic information. In metaphase, chromosomes line up along the cell's equator. This arrangement ensures that each new skin cell will receive an identical set of chromosomes when the cell splits. In anaphase, spindle fibers pull sister chromatids apart, moving them to opposite ends of the cell. This separation ensures that each new cell inherits an identical and complete set of chromosomes, enabling it to function properly as a skin cell. In telophase, chromosomes decondense, becoming less visible, and new nuclear membrane form around each set of chromosomes. This restoration of the nucleus ensures that the new skin cells can properly manage genetic material and regulate cell activities. In cytokinesis, the cytoplasm divides, creating two separate daughter cells, each with its own nucleus and organelles. This final step produces fully independent skin cells ready to integrate into the skin tissue, supporting its continuous renewal and repair process. First, DNA helicase unwinds the double helix, creating a replication fork. Single-strand binding proteins stabilize the separated strands, preventing them from rejoining. Unwinding the DNA allows cells' replication process to access the nucleotide sequences and copy the DNA. Then, RNA primase synthesizes a short RNA primer on each DNA strand, which provides a starting point for DNA polymerase. DNA polymerase synthesizes a new strand by adding complementary nucleotides to the template strand. The leading strand is synthesized continuously in the 5' to 3' direction, while the lagging strand is synthesized discontinuously in short fragments called Okazaki fragments. The complementary base pairing ensures accurate copying of genetic information, essential for the function and identity of the new skin cell. Then DNA polymerase replaces the RNA primers with DNA. DNA ligase seals the gaps between Okazaki fragments on the lagging strand. This step completes the synthesis of the lagging strand, ensuring a continuous and complete DNA molecule.